My name is Angelica Ramirez, and today I'll be presenting on humoral immunity. So the phases and types of humoral immune responses are: first, naive B lymphocytes recognize antigens. Then the helper T cells activate B cells to proliferate, leading to clonal expansion. Then they differentiate into antibody-secreting plasma cells. Some of the activated B cells undergo heavy chain class switching and affinity maturation, and some of them become memory cells. On stimulation of B lymphocytes by antigen, we have two. T-dependent antigens are when helper T cells help B cells respond to only protein antigens because T cells can recognize protein peptides due to MHC. The T-dependent antigens stimulate the generation of long-lived plasma cells and memory B cells. The T-dependent responses are specialized and long-lived that involve protein antigens and under the influence of helper T cells. On the other hand, T-independent antigens are multivalent antigen agents like polysaccharides, lipids, and nucleic acids that can stimulate antibody production without the involvement of helper T cells. T-independent responses are simple and involve only the direct activation of B cells by antigens. The role of innate immune signals in B cell activation. B lymphocytes express a receptor for a complement system protein that involves secondary signals for the activation of B cells. When the complement system is activated by a microbe as a part of the innate immune system, the microbe becomes coated with fragments of protein C3. C3 has a fragment called C3d, and B cells express a receptor for C3d, which is CR2 as a complement receptor. The B cell recognizes the microbe's antigens by their BCRs and also recognizes the bound C3d with the CR2 receptor. The engagement of CR2 receptor enhances the antigen-dependent activation responses of B cells. Microbial products can also directly activate B cells by using PAMPs. B lymphocytes have various toll-like receptors. PAMPs bind to the TLRs on the B cells that trigger activating signals. The combination of signals stimulate B cell proliferation, differentiation, and immunoglobulin secretion that promotes antibody responses against microbes. On the heavy chain isotype class switching, different antibody isotypes have different functions, so the process of being capable of isotype switching broadens the capabilities of the humoral immune responses. Heavy chain isotype switching is induced by a combination of CD40L mediated signals and cytokines. Antigen stimulated B lymphocytes differentiate into IgM antibody secreting cells, or with the help of cytokines and CD40L, and some B cells can differentiate into cells that produce different immunoglobulin heavy chain isotypes. Cytokines produced by follicular helper T cells determine which heavy chain isotype is produced. IgG antibodies opsonize microbes and promote their phagocytosis and intracellular killing, and IgE is stimulated by IL-4 that eliminates helminths. IgA provides mucosal immunity. Affinity maturation is when the affinity or the strength of antigen-antibody binding of antibodies produced in response of a protein antigen increases with the repeated exposure to that antigen. So if the antibody is exposed to an antigen several times, the binding between both increases. Affinity maturation occurs in the germinal centers of lymphoid follicles. The regulation of humoral immune responses. Antibody feedback is a process by which antibodies bind to antigens, inhibits further antibody production. Antibody feedback serves to terminate humoral immune responses once enough IgG antibodies have been produced. The effector functions of antibodies. IgG is in charge of neutralization of microbes and toxins, opsonization of antigens for phagocytosis by macrophages and neutrophils, the activation of the classical pathway of complement, the antibody-dependent cellular cytotoxicity mediated by natural killer cells, neonatal immunity via the placenta, feedback inhibition of B cell activation. IgM is in charge of the activation of the classical pathway of complement. IgA provides mucosal immunity and neutralization of microbes and toxins. And finally, IgE is the eosinophil and mast cell-mediated defense against helminths. Vaccines is the process of stimulating protective adaptive immune responses against microbes by exposure to non-harming forms of components of the microbes. There are many types of vaccines that are in use, such as live attenuated or killed bacteria, which functions as an antibody response. Live attenuated viruses protects against antibody response, cell-mediated immune response. Antigen vaccines give an antibody response. Conjugate vaccines help a T-cell dependent antibody response to polysaccharide antigens. Synthetic vaccines give an antibody response. Viral vectors, cell-mediated and humoral immune response, as well as DNA vaccines also provide cell-mediated and humoral immune response.